mentioned, just like with the lecture. If you're here physically, just going to write down all the attendance, or if you're joining us online, if you're live streaming this or you're here with us, write down all the attendance words as I say them. At the end of it, send me an email that has a list of attendance words, in addition to one question about the material. Now, if you're watching the video at a later date, so if you're not live streaming this, then you're going to also include screenshots every time I say the screen word. And like I mentioned earlier, for Edwards, I wrote down who's here physically. But of course, for points, it doesn't matter if you're here or not. So Edwards, I know that you're online, but you'll get your points once you send the email with the attendance word. So the first word for attendance, nice and easy, it will be lab. Lab is the first word for attendance. So if you're watching the video, none of you who are watching right now, if you're watching the video at a later date, your screenshot should include a picture that shows that word lab that's circled. So let's jump into it. I really don't want to keep you here long. I want to get through this as quickly as possible, but also if you have questions, make sure you slow down and let me know. As I already mentioned, today's lab is based on attendance. This is actually old. So it used to be you just had to be here. Now I'm requiring you to send attendance words. Also, on the other hand, if you come in late, you will still get half credit. For anybody who comes in late, of course, they don't hear me talking right now. Or if you leave early, you only get half credit. Of course, you could also watch the video later to get full credit. Next week's lab grade is based on what it usually is. We've got that 5 points for the pre-labs that's due this Friday, 15 points for the lab worksheet, which adds up to 20 points. But then again, which is what we're going to talk about today, you'll also have a lab report that's due 2 weeks after you get your graded lab back, and that's worth 20 points. So here's some questions that we're going to answer in this lab. Does the rate of photosynthesis increase as light intensity increases? So basically, as things get brighter, do plants and other photosynthetic organisms do more photosynthesis, so to speak? That's one of the experiments. Another question is, do photosynthetic organisms not photosynthesize, but do they respire when there is no light? Then we're going to ask, does an increase in temperature result in an increase in the rate of photosynthesis? In other words, does temperature affect the rate of photosynthesis? And then we'll ask, does temperature affect the rate of respiration? And then finally, we're going to ask, which colors of light affect photosynthesis, result in more photosynthesis? So those are the questions. We're going to talk about all these separately. So here we go. Question is, how are we going to answer these questions? So for example, let's say, forget what you see there, and hopefully you've already looked at this, but it's okay if you haven't. But if you have already looked at it, pretend you haven't. If we had all semester, for example, to figure out which colors are best for plants when it comes to photosynthesis, what kind of experiment could we do? If we had the full semester to do it, what would you do? Like if we started it on day one and then collected our last data on the last day of class, what's an experiment we could do to see which color of light is best for plants, so to speak? Yeah, basically, right? We could set up different groups of plants, grow them under different colored lights, and then maybe just take some measurements. What could we measure if we had full semester? What would our measurements be? The what? The length, right? How tall it is. What else?
Like the leaves go.
Yeah, the leaves, right? Maybe its weight, its width, the number of fruit it produces, whatever. But we don't have all semester, right? So we have to figure out a different way. We have to figure out a different way to measure the rate of photosynthesis and respiration. So how are we going to do that? Not only do you need to know it, like you always need to know how we do these procedures in an experiment, but this time you really need to do it because you have to write a lab report about it. So before we talk about how, let's understand basically how photosynthesis and respiration works. So photosynthesis uses water and carbon dioxide to produce glucose and oxygen, right? Meanwhile, respiration is the opposite. Respiration uses glucose and oxygen to produce carbon dioxide and water. So without jumping into specifically how we're going to do it in our experiments, logically, very basically, here's how we could do it. For photosynthesis, if we can measure the amount of water, if the water level was going down, we could say photosynthesis is happening because it's using water. If the levels of carbon dioxide were going down, we would know photosynthesis is happening because it uses carbon dioxide. If we could somehow measure the levels of glucose, if that was increasing, we would know that photosynthesis is happening because it's producing glucose. If we can measure levels of oxygen, if it was going up, we would know that photosynthesis is happening because it produces oxygen. Of course, everything I just said would be the opposite for respiration, right? If glucose levels are going up, going down, respiration is happening. If oxygen is going down, respiration is happening. If carbon dioxide is going up, respiration is happening. If water is being produced, respiration is happening. No, excuse me, I think I may have misspoke earlier when I talked about photosynthesis. If water is being used, photosynthesis is happening. So it's the opposite. If we could somehow measure any of those, that's how we can measure photosynthesis and respiration. And there is a way we can measure some of those. Here's a little background information. You're going to need to know this for the pre-lab, questions. But a little something you might not know is that when you mix carbon dioxide and water, it produces something called carbonic acid. Now, for the lab report and for the lab worksheet, sorry, I'm just looking to see what that noise is about. I don't see anything. Making sure everything's okay here. Okay. Anyway, sorry. I wanted to make sure everything was okay. It is. When I hear those noises, I get paranoid. So yeah, carbon dioxide and water produce carbonic acid. For the lab report, for the lab worksheet, it's not really important that you know it's carbonic acid, but you definitely need to understand that when you mix carbon dioxide and water, it produces acid. So that being said, now that you know this, right? If carbon dioxide and water produce acid, then which of these two is happening if the levels of acid are increasing? If we're producing acid, because this is what we're going to measure. We are going to measure the amount of acid. If the amount of acid is going up, which one of these two is happening? Photosynthesis or respiration? Don't overthink it. If you just look at it really basically, it's actually quite simple. We're producing acid. Let's take a different step back. If we're producing acid, let's just start with that. If we're measuring the amount of acid and it's increasing, what are we producing? Are we producing water, carbon dioxide, glucose, or oxygen? If the levels of acid are increasing, which of those four things are we producing? Water, carbon dioxide, glucose, or oxygen? Actually, just forget water, just to make things simpler. Carbon dioxide, glucose, or oxygen. If we're producing acid, which one of these three things are we making? Carbon dioxide, glucose, or oxygen? Perfect. Carbon dioxide. We're producing carbon dioxide. So knowing that we're producing carbon dioxide, then which one of these is happening? Photosynthesis or respiration? Respiration, right? Because respiration produces carbon dioxide. Perfect. So you guys figured this out on your own, basically. Therefore, photosynthesis reduces the amount of carbon dioxide, right? Because we know this. Photosynthesis uses carbon dioxide, right? So if it's using up carbon dioxide, that's going to reduce the amount of acid, right? We're going to have less acid. And if we have less acid, that means the pH is going to go up, right? Meaning it's less acidic. So all that long-winded stuff to say, when pH goes up, photosynthesis is happening. That's the most basic thing of what you need to know to do these experiments. For the lab report, to truly understand it, you need to know all these other things you said. But just to sit there next week and understand what's happening, that's what you need to know. When pH increases, photosynthesis is happening, right? Because photosynthesis is using up carbon dioxide, therefore it's reducing the amount of acid, which makes it less acidic, aka more basic, aka the pH goes up. Is everybody clear on that? I'm hoping that's pretty simple. And of course, the opposite would be true of respiration. Respiration, as we already mentioned earlier, like Noah basically told us, respiration is going to be producing carbon dioxide, which is going to produce carbonic acid, which makes the pH go down. So again, all that technical stuff, which is good for you to understand for when you're writing your lab report. And of course, it's going to help you during the exam as well. But the most basic things you need to know next week to understand what's happening, we're going to measure the rate of photosynthesis and respiration by the pH. If pH goes up, photosynthesis is happening. If pH goes down, respiration is happening. Now, if it stays neutral, as far as we're concerned, I'm not going to talk about this much, but if the pH doesn't change, basically what's happening, most likely, is that photosynthesis and respiration are happening, but at the same rate, right? So as photosynthesis is happening, it's producing glucose and oxygen. But meanwhile, these things, these algae beads that I'm going to tell you about later, they also go through respiration. Everything does to live. So it's also using the glucose and oxygen. It's just doing it at an equal rate. So that's something else to keep in mind when we're going through these experiments. So when we say photosynthesis is happening, when the pH goes up, what we're really saying is if photosynthesis is happening respiration, and vice versa. I keep getting that noise and then I don't see anything. All right, so Edwards, I see that you're online, but if you have any questions, just take yourself off mute and ask your question. That way I don't have to keep going back and forth. But anyway, hopefully that's just a glitch. So any questions about this concept? This is how we're going to measure photosynthesis, change of pH. All right. Next, get it pulled up. There we go. So how do we do that? We're going to have these cuvettes, and luckily for us, he already brought them in because they don't look exactly like that, but they look similar. Instead of being round, they're these little cube-looking things, hence the name cuvette. But other than that, they're the same. They have little algae balls in them, which you might or might not be able to see when we're done. You feel free to look around in this cart. And they have liquid in it. That liquid includes water, obviously, because they're going to need water to live. But it also has pH indicator. So this liquid will change colors as the pH changes. So what we're going to do, whatever experiment you're doing, there's going to be different details, but basically all you're going to do is put these little cuvettes in different conditions, depending on which experiment you're doing. And every 5 minutes, you're going to take it, you're going to compare it to this strip of paper right here to determine its pH, and you're going to write its pH. Every 5 minutes for 45 minutes. That's it. We're just going to track its pH change over 45 minutes. And that's going to tell us how quickly or slowly photosynthesis or respiration is going to happen. So here's a question for you, though. Why do this every 5 minutes, right? Why not just take the pH at the beginning and take the pH at the end? Any ideas why we're doing that, other than busy work, because you're students and we have to keep you busy? Actually, I made that up. What is the real reason? Why do you think, why would we need to take it? Or instead of that, forget about the every 5 minutes thing. Let me reword the question. Why take it periodically instead of just once at the beginning or once at the end? Why have more than 2 measurements? What is it?
More than one.
Yeah. I think you're getting there. And you're right. What I'm getting at here, what I'm trying to get at is, well, this is very simple. But like take, for example, if you have 2 different conditions, right? You got one in a lot of light and one in a little light. What if they both start at the same pH and end at the same pH? Then, if that's your only data, you'd be like, oh, they both photosynthesize the most. But if you have data every 5 minutes, one of them, like you try to imagine on a graph, maybe one of them really peaked off really quick and leveled off, and the other one eventually got there. So you would know the one that leveled off first photosynthesized. Maybe there's just a limit to how high the pH can go, as there should be, right? Because remember what we've learned about enzymes. Enzymes are very sensitive to their pH. So there might be a point here where the pH gets so much that these enzymes doing the photosynthesis or the respiration stop working. So again, this is why we need to take those measurements every 5 minutes as opposed to once at the beginning or once at the end. Because you might need that in between data. Hopefully for you guys to make things easier, hopefully it's not like that. Hopefully the beginning and the end is different for each one and the data is easy to interpret. But I doubt it. All right. So again, we got these cuvettes filled with pH indicator, which is a liquid. They also have algae beads. And again, these are the questions we're going to ask. I'm not going to repeat all that because we're going to talk about them separately here in a second. Yep, let's talk about them each individually. So here we go. The first one. Let's see. Do photosynthetic organisms respire faster and more intense light? Right. Another way of asking that is, does light intensity affect the rate of photosynthesis? So you're going to have 4 cuvettes. We're going to have these lamps here, right? You're going to set them up on this table. One of them is going to be very close to the light. One of them is going to be farther away from the light. One of them is going to be even farther from the light. And one of them is going to be kept in complete dark. For every 5 minutes, we're going to check their pH, write it down on the table, and at the end, we'll calculate the total change to see which one was photosynthesizing the most or the fastest. Does that make sense? Pretty simple stuff, right? The next one's even simpler. If you get experiment 2, you kind of got lucky. And it's all going to be random because this one's really easy. So this one, we're just asking, does respiration even happen? Respiration, not photosynthesis. Do photosynthetic organisms respire in the dark, right? And you're going to have one in the light, one in the dark. You're going to want to see if the one and the dark decreases, indicating that respiration is happening. Yeah, and it's that simple. What do you guys think, though? Do you think they will respire in the dark? And I should have asked for the last one, but and this is going to help you too, because soon when you do your pre-lab lectures, you're going to have to answer the question I'm asking you right now anyway. So might as well get it in your head, answer it in your head. So when you do the pre-lab, you can answer it and write. What do you think? Do photosynthetic organisms, in this case algae, do they do respiration in the dark? Not photosynthesis, but respiration. What do you guys think? Someone, just one person give me an answer before we move forward.
Yes.
Yes, good hypothesis. I agree. Otherwise, they wouldn't survive, right? But something to keep in mind too, because I'm not an algae expert, they're going to be doing this for 45 minutes. And what do we need for respiration? Glucose. Where do we get glucose? Photosynthesis. So there might be some point, and I don't know if it's going to be within these 45 minutes, but logically, these are things you kind of need to think about when you're doing these experiments. Eventually, you're going to run out of fuel, which is glucose and oxygen, right? So without that light, eventually those algae are going to use up all the glucose and all the oxygen that they produced. So your hypothesis is a great one. That would be my hypothesis too. Yes, photosynthetic organisms respire in the dark, but then a caveat, eventually they do need light, right? Not that you'll need to write that down for the lab report or your pre-lab, but yeah, it's just something to consider. Obviously, if plants would just die at night, right? If they required light all the time, they would just die at night. But they don't, because they've stored up glucose in the form of starch, as you already know, and of course oxygen they get from the atmosphere. All right, the next question we're going to ask is, does an increase in temperature result in an increase in the rate of photosynthesis? In other words, how does temperature affect respiration? Photosynthesis. And some things you've already learned should help you formulate this hypothesis. For example, you learn from enzymes that generally speaking, the warmer things go, or the warmer things get, the faster chemical reactions happen, right? So by that logic, the warmer things are, the more photosynthesis you should expect to happen. But there are some things to consider. For example, if it gets too warm, remember you learned that they can be denatured because enzymes are very sensitive to their environment. So what if it gets too warm and they start to denature and they start working less? Another thing, we don't know anything about this algae. I don't anyway. Somebody does, I don't. As far as I know, these algae might grow on the bottom of icebergs, right? So if they're used to that environment, if they're evolved for that environment, then they might do better on ice, right? Of course, on the flip side, if these algae came from some tropical pond from, South America, where they're getting lots of heat, they're going to be evolved for that. So anyway, anybody want to tell us what your hypothesis for this? Do you think as temperature increases, do you think it'll photosynthesize more or less? What do you guys think? Just one hypothesis from one person, and we'll move on.
Yes.
All right, we'll go with that. Yes. And again, I'm asking you this, but you're going to have to write it down when you do your pre-lab. So I just want to say it out loud if you want. The other experiment is going to be the same thing, except we're going to be looking at respiration instead of photosynthesis. But the logic should still be the same, right? Generally speaking, the higher the temperature, the quicker chemical reactions happen. However, specifically, things are evolved for different things. So just some stuff to keep in mind. I won't even ask your hypothesis on this because, like I said, it's basically the same thing as the photosynthesis experiment. We're going to have one cuvette on ice, right? This thing contains ice. You can have one on ice and then one here at the edge. And you're just going to check its photosynthesis every 5 minutes. Anyway, any questions about that? All right, the last one. I don't know why, but this one's my favorite. I always use this as an example. It's basically which color is best for photosynthesis. And you're going to learn more about this when we get to that chapter. But I don't know if we're going to get there in time for this, so let me go ahead and give you a few quick lessons. White light is all light. So we're asking about green, red, blue, all that. Any color you can think of is included in white light. This is why when you hit white light on a prism, you get a rainbow, because you're breaking up all the lights that are already there into their respective parts. So that's something to keep in mind. Another thing to keep in mind, plants are green. The reason they're green is because all that white light is hitting them, all the colors, and it's absorbing all of those colors except green. Green is the one color that's being reflected back and hitting our eyeballs. So that being said, which color do you think, based strictly on what I said, which color do you think would be best for photosynthesis of these? White, green, red, or blue? White, I agree. Logically, that makes sense because that's all the colors. And which one do you think might be the worst? Green, right? At least based on what I described, that makes sense because that's the one color that's being reflected. There are some other things to consider, like for the fact blue light is very high energy light, red light is a very low energy light. All these things you'll learn about later, but that's the basics. So if you know that, I should help you with this. Also, if I can do it, if we can do it, if we can make this happen, we can get more cuvettes and get some other colors. I'd be interested. Like I'd throw an orange light in there or a yellow light if we can. It's not necessary, but if we can, let's do that. So that's it. That's all the experiments we're going to do. I mean, that's not done. That's not it for the lecture, unfortunately. Don't get too excited. But for the experiments next week, you're all going to break it down into 5 groups. Well, 6 groups, because I'm sure there's going to be at least one person who didn't do the pre-lab and can't do any experiments. But anyway, those other 5 groups, each group is going to do one experiment. Just like last time we had a similar lab, when you do the lab worksheet, you're going to do a lab worksheet for the entire lab work, regardless. You do all the experiments in the lab worksheet. Your lab report will be on the one experiment you did, and your graph on the lab worksheet will be on the one experiment you did. That makes sense. Okay, and of course, I'll be there to help you next week to remind you that. Of course, you can do extra credit if you do a graph for each one of the 5 experiments. Here's another thing I want to point out, because people, I really get this. I can tell students get this confused when I read the lab reports, which is why I love lab reports. They're a pain in the rear end to grade. They're a pain in the rear end to write, but it really helps me learn what you've learned. But just to be very clear, this is not a pH experiment. This experiment is not about pH. pH is merely the tool. It's what we're using to measure the rate of photosynthesis, right? So please don't think like this is because, we just got finished not too long ago doing a pH experiment. But no, we didn't, did we? We should have, but we were snowed out, so you did it virtually. Anyway, the point is, this is not about pH. This is just a tool. How else can we measure it? We've already talked about it, right? So I'm not going to talk about it too much. I already said something like, if oxygen is going up, for example, we know the photosynthesis is happening. If glucose is going down, respiration is happening. Measuring a tree, how tall does it get? All those things we've already talked about, that's how we can measure. And I'm not just saying this for nothing, because these are the questions that you'll be answering on the lab report, some of them. You're going to need to talk about, like, here's the experiment we did, here's the results, and then after that, you're going to talk about, here's some other experiments we could do to test our hypothesis. Some other things to keep in mind, in theory, no cuvettes in the dark should increase in the pH, right? Because in theory, if they're in the dark, they're not photosynthesizing. Therefore, in a sense, you could argue that if your pH did increase, all your results are invalid. Which one? You won't get in trouble for that. You won't lose points. It's not your fault if something goes wrong. But yeah, it's something to consider. Would you say that? Like, that is something worth considering. If the pH goes up when they're in the dark, something must have went wrong. Better example, though, I should change this. No, we'll just leave it at that. Anyway, the point is you should just keep these things in mind. If something seems wrong about your data, then it's okay when you write your lab report to be like, according to the data, this, and this, but there's some red flags here. So even though my hypothesis was supported, I don't think this data is enough to say that it's true. Whatever. We'll cross that bridge when we get there. Again, as should be the case, which is why you keep getting these questions every week, is because I've been leading you up to this. As we're doing these experiments, you should think about how you can improve the experiment. Not the worksheet. I'll always ask you about that later too, but scientifically, how can we improve this experiment? Right? So think about that as we're doing the experiments. Again, think about other ways to test your hypothesis. Because in real science, you don't just do one experiment. Usually, they'll be like, oh, my hypothesis was supported or not supported, I'm done, right? You come up with other ways to do it. And we've already talked about some of them, right? Growing a plant for a semester and measuring its height. Whew. So that's it for the experiment. Let's really quickly talk about how to write a lab report. Before I even do that, first of all, I want to go ahead and tell you the practical stuff. Then we'll talk about this more philosophical stuff. But anyway, practically, what I'm going to do is I'm going to give you a form. It's almost like a rubric. Have you guys seen a rubric? And usually it's like a table, and you get like, oh, if you get this far in the table, you get X%, this far, so on. This is going to be more straightforward than that. It's going to be straight up. And if you read the rubric from the beginning to the end, you have no problem. It'll say question number 1, not question, let's say 1. Is there a title? This is my grading rubric. Is there a title? Parentheses 1 point. So you're going to lose a point if you don't have a title. 2, is the title appropriate for the lab report. You get a point for that, right? You just go down step by step. If you just follow step by step, it should be easy. Anyway, first question you might be asking is why? Why do you have to write a lab report, right? None of you are biology majors, so why do you have to write a lab report? The most simple answer is because I told you so. Not really, but sort of. But it is part of the course, and it's not really because I told you so, but because someone told me so, right? Someone says, even for non-biology majors, you do need at least one lab report in the science course. So there we go. That's the most basic answer because we have to. Another thing I love about it is these two parts too, or the second part. To truly understand the experiment, you have to be able to understand the experiment to write a lab. You can do a lab worksheet and not truly understand what happens. Just take the data, answer the questions. But to be able to write a lab report, you truly have to understand it. And again, in a perfect world, I would like to do a lab report for every lab instead of a lab worksheet. But that would be too much work for you, too much work for me, and it's only 100 level class, so we don't do that. Another thing, and this is a little bit less important because this doesn't help that much, but it should also help you understand research articles, right? And hopefully that will be important later in your life because as college-educated citizens, hopefully you don't just accept what people tell you. Hopefully you look for their information. Oh, you said this? What are your sources? And then look at the sources. But to be able to understand those sources, you need practice. And one of the ways of practicing is writing a lab report. Because a lab report is like a really light version of a peer-reviewed article. Very light. But you're going to understand those processes, so that should help you understand peer-reviewed articles. So there's some things to consider. Do not use the internet. What's a little start with? I'm going to start and put myself like, hey, don't use the internet. Oh yeah, I remember now. So when I say don't use the internet, what I mean is this. I'm going to give you the steps you need to take. I'm going to break it down. First, you can do an abstract, an introduction, blah, 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 but I'm about to tell you. My point is, if you were to Google, how do I write a lab report? You're going to get all different answers. Just like a peer-reviewed article, each peer-reviewed article, whether they're talking about heredity or Scientific American, whatever it is, they have their specific rules. Like, all right, if you're submitting your work to us, I want you to do it in this format. And then some other peer-reviewed article talking to the same scientist who's submitting the same work would say, all right, I want you to submit this in this format. So I'm telling you the format that you need to submit it in. So do not Google it. Do not look up how to write a lab report. It will not work. You'll lose a lot of points. Again, like I already mentioned, you only need to write about the one experiment you did. And I know this sounds weird, but you will lose points if you do more, right? And that's one of those few times when doing more work would hurt you. Only write about the one experiment you did. Now, if you want to, you can write separate lab reports for extra credit, but don't add them all into one lab report, right? For the one lab report, just do one experiment, specifically the one that you did. Write about it in past tense in the first person. So say the first thing I did was put this cuvette 25 centimeters from the light. Then I put this cuvette 35 centimeters from the light, right? So first person, past tense. It's going to be double spaced. It's easier to read. There is no minimum. I always get this question. What's the minimum? How many words? How many pages? There is no minimum. Short and sweet is what I like. Just get to the point. This is scientific writing. You don't need to use a bunch of adjectives. And I gently put the cuvette on the table and wondered what the results would be. And then I grabbed the other one gingerly. And I don't do that, right? Straight to the point. Yeah. Like I said, I'm going to give you a rubric. Just go by the rubric. No need for extra stuff. If I do need more from you, I'll leave you feedback. Like, hey, look, you lost some points because I felt like you could have written a little bit more about this. And then just go back in and write a little bit more about that, right? So short and sweet. If I need more from you, I'll tell you. Because this, just like almost everything else in the semester, is not set in stone. I'm going to give you a grade. It doesn't mean you can't go back and improve. Due date. 2 weeks from the day you get your grade back. Right, so your lab report will be due on Friday. Your lab itself will be due next Friday, past due next Friday. Let's assume by some miracle I had nothing else to do and graded it that day, then 2 weeks from that day it will be due. But again, 2 weeks from when you get your grade back is when your lab report is due. And I'll tell you, when I give you your grade back, or the feedback will be, blah, 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 good job. Your lab due date is, and I'll give you the specific date. But as you're going to be able to see when I'm telling you how to write a lab report, you can write a lot of your lab report before you even do the experiment. A lot of it. You just write it. You can even, in a sense, you can write all portions of it and then just fill in some blanks later. But at the very least, you can write, well, you'll see. Anyway, the point is, You don't have to wait until you get your lab back to do your lab report. Anyway, let's talk about how to write a lab report. Let's talk about some things you lose points for. And I'm not that picky about this, but it should be free of grammatical, spelling, and typing errors. And I'm not that picky. I'm not the best when it comes to grammar anyway. I'm just going to be looking at things that if it sticks out to me, who's not that great at it anyway, then you're going to lose points. Like, I don't know why, but for some reason, the word seen, that's a pet peeve of mine. So, well, I've seen that it changed pH. I'm like, that's not right. Or if you keep starting, you just start in sentences with lowercase, like really basic stuff. If you're making basic errors, you're going to lose points. That being said, and this is a new thing, relatively new, do not let AI write your lab report. Don't even let it rewrite your lab report. Because again, even though I'm saying you're going to lose points for that, I'm not that picky. So don't feel like it has to be perfect. An AI could screw it up. So, and AI is a good tool. Sometimes it is. I use it to rewrite some things too. But I just want for now to you not to do that, so I can see how you write. Right? This is all about helping you learn to write. And again, it's a good tool, but let's just not use it for this assignment. It should make sense. This is a big one. This is the test that it should pass for you. You should be able to give it to somebody who's not had this class and say, hey, read this, does it make sense? There should be enough information for every little thing that you have in there makes sense. For example, and you're going to learn about this here in a second, in your introduction, you're going to explain all that pH stuff that I just explained to you. But if you were to fail to do that, then when you give that lab report to somebody and they read about the change in pH, you're like, what the change in pH? What does that have anything to do with photosynthesis, right? If your lab report makes sense to somebody who's not had this class and didn't do the experiment, you probably did a pretty good job. It should be accurate and correct. You will lose points if you say things that were inaccurate, like, well, the pH went up, so I know respiration happened. No, that's the opposite of true, right? So you lose points for that kind of stuff. So just be careful. It should be precise. We don't really have it. That's more for a bigger, higher level class. That's when you're doing like averages and how many decimal points. So yeah, I don't know. Let's not even worry about that. It should be succinct. Again, like I said, you don't need to be writing a bunch of words, a bunch of adjectives and adverbs. Straight to the point. It should be clear that you understood the experiment. That's a big one too. This is another one of the huge ones I'm looking at. If I'm reading it and like you're giving me all these results, like, yeah, the pH increased a lot for the color blue. But then you go on to say, so yeah, the color blue was the worst photosynthesis. Like, I don't think you understood that. You just told me that pH increased the most for blue, indicating that photosynthesis happened the most. Right? So that's the kind of stuff I'm going to be looking for. Which is a good point, good time to remind you. If you didn't understand the experiment, that's okay. You're not here to just know everything. You're here to learn. So if you didn't understand it, even after you did it, I can talk about it and explain it to you. So don't feel like when you sit down to write your lab report a week after you've done the lab, you're like, man, I don't know if I really understand that. I don't feel like I can't ask them because that's going to indicate that I don't know what's happening. It's okay if you don't know what's happening. That's what I'm here for. It's to make sure you understand what's happening. So if you don't understand the experiment, let me help you. So any questions about this slide? All right. Another thing I want to point out really quickly, because again, I don't want to waste a lot of your time. This PowerPoint that we're looking at right now is on WBSU Online. So you have access to this. That's also by taking a picture, which is smart, but not needed. So again, you're going to have a little rubric. The rubric's already up there too. You can pick up this, you can open this document right now and look at it. This is what it looks like, a portion of it. That's just the beginning. But like I said, this is how straightforward it is. Is there a title? 1 point. Is it appropriate? So for example, if you put lab report, that's not appropriate. It needs to specifically talk about the experiment. Anyway, I'm not going to go through each one of these, but this is how simple it is. If you just go from the top to the bottom and make sure you address each one of those points, you should be fine. All right, here's where it gets a little bit more complicated. Again, do not Google this because every source is going to be different. Here's how I want you to do it. You're going to have a title, and we're going to talk about all these separately. A title followed by an abstract, followed by an introduction, followed by materials and methods, followed by results, followed by discussion, figures and tables, and literature cited. So that's a good, we'll talk about that. Title, really quickly. I've already mentioned most of this. You're going to, it's 1 of 5, not 4. You are going to do a specific experiment. That's the one you're going to write about. Therefore, your title should indicate which one of those experiments you wrote about. Make sense? So again, photosynthesis respiration lab, not a good title. You will lose a point for that. The effect of color on the rate of photosynthesis would be a good title if that's the experiment you did. So any questions about the title? All right, abstract. This one's important and good for you guys to know about because, again, as a college-educated citizen, once you're done, hopefully you don't just accept what people tell you. Hopefully you look up things, right? You're a critical thinker. And a big part of doing that, looking up peer-reviewed articles, is this abstract. Very often, an abstract's going to give you all the information you need to know if you even want to read that paper. And it's going to give you the basic results. Very often, when people claim something, they just claim, they say, according to this study, they give you a very basic statement, even though it's a really long peer-reviewed article. That's what the abstract is. Right? That abstract is like, here's basically what we did. Here's basically the results. There you go. And that's what you're going to do. This is a brief summary about what the paper is about. Again, it should be labeled, so it'll literally say abstract, and then you're going to write your paragraph. It should let the reader know if they want to read the rest of the paper. Of course, in this case, practically, I'm going to be the only one reading it. And yes, I have to read it. So we know in actuality, that's not the case. But in the real world, an abstract, again, it's like, here's basically what we did. Here's the results. So, some scientists do need to read the entire thing, and that's what they're looking for. You should include the question being answered in the lab. So again, if you're doing the light experiment, like, does the color of light affect the rate of photosynthesis? It's the question I was answering in this lab. And we put, you get a very basic description of what you do, right? So we put photosynthetic organisms or algae or whatever you want to say in different colors of light. And it turns out white was the best color or blue was the best color, whatever it may be. Get very basic description of what you did, very basic, because you're going to get in more detail later, and just the very basic results. Any questions about the abstract? All right, introduction. This one's a little bit more important. Like I said, your whole lab should make sense. And let me use some examples. For example, and we're going to talk about this obviously here in a second. Results are strictly going to be this pH started here, ended here. pH of this started here, ended here. That's all you're going to say for the results. But without the introduction, those results wouldn't make sense. This is why you have the introduction. So when the results, if you just talk about the change in pH, that would make no sense to the reader. Well, luckily for you, with the introduction, you explained what the pH changes mean. So when you talk about it in the results, the change of pH, you don't need to explain it. You've already explained it. That being said, let's talk about the introduction. And again, this is all going to be provided to you in the rubric. It has to be labeled just like everything else. It'll say introduction, and then you'll write your paragraph. It should give enough information about photosynthesis and respiration that the reader will understand the rest of the lab report. So again, the change in pH is a must. You have to explain that. But of course, to explain the change in pH, you also need to explain the fact that photosynthetic organisms use carbon dioxide, which is going to change pH, and photosynthetic things, organisms that are respiring are producing carbon dioxide, right? So all that information, the basics, you need to explain. Also, depending on what lab you're working on or which experiment you did, you might need to explain something about light, right? Like the color of light or light intensity, or maybe a little bit about temperature. Like, oh, generally, the warmer things get, blah, blah, all the stuff we've already talked about. That's the information you want to put in there. Because again, you want to have enough background information that the person can read the entire lab report and it'll make sense. Of course, you're going to, again, I know this is going to be very repetitive, and you've already been doing this in your lab worksheets. I've been building you up to this. Once again, you're going to repeat the question. In this experiment, I was trying to find out blah, 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 right? So you're going to repeat it. This is the first time, though, that you're going to mention your hypothesis. Here's the question that we were trying to answer, or that I was trying to answer. Here's my hypothesis. Here's why I think. Right? So just like the lab worksheet, you're going to be answering these questions in your pre-lab. If you do it properly, you should be able to just copy and paste it into your introduction. And yeah, like I already mentioned this, right? You should explain how photosynthesis and respiration change the pH. That way, the stuff later in the lab makes sense. There's a lot of things you should not include. So this is another big mistake people make. You don't need to talk about how you did the experiment yet. That's the whole different section for that. You definitely don't even mention the results because you gave a basic description in the abstract, and then later you're going to talk about it in more detail. But for the introduction, you don't need the results. You don't need the specifics of how you did the experiment, just the basics. Any questions about the introduction? All right, materials and methods. This is probably the easiest. You're just going to write about how you did it, right? This section can give enough information that the reader could perform the experiment from reading it. That should be your goal, your guideline right there. If you can write it in enough detail where someone else can do the experiment, you've done it fine. Right? You should mention everything used. And so we had algae beads that were inside cuvettes that were filled with water and pH, see the pH indicator. And we had a pH strip to compare it to. Like everything you used, you need to include it. That's usually the biggest mistake if people forget a part or forget a material. Another thing they do sometimes is forget a method. So make sure you write down everything that you did. Now, granted, you don't have to say like you could. For example, you could say every 5 minutes we check the pH. But you don't have to say we check the pH to begin with, and then in 5 minutes we checked it again, and then at 10 minutes we checked it again, and then at 15 minutes we checked it again. I don't need to do that, but yeah, you do need to write down everything you do and everything you used. It should not be copied word for word from the lab manual. I mean, none of it should be, except with, like I said, some of your answers and the lab worksheet. You can use those, like the hypothesis, stuff like that. But you do not want to copy your methods. For one thing, it's not written in first person, right? Your lab report, your lab worksheet is not written in first person. Yours needs to be. This was a big thing with that. I think that's it. Yeah. Oh yeah, another thing. Write down in your lab report what you did. So you're going to see when we do these experiments, these are your experiments. We have this guideline, and it's almost like a recipe. We can change it. We probably will make some changes. Well, yeah, I know it said to do this, but let's change it to this. Right? We might make some changes to that the experiment design, which is okay. But when you write about it, you write about what you actually did, not what you were supposed to do, so to speak. That make sense? All right. Any questions about that? Wait, there's a little bit more. I'm sorry. Yeah, again, your introduction should explain why you're doing things the way you're doing it. So you don't have to explain why you put a cuvette 10 centimeters from the light, one 25 centimeters from that. You've already explained it. You just say, I put one here, put one here, put one here. I check the pH every 5 minutes. Right? All right, next are the results. This is where people get a little bit confused. And again, I've been leading you up to this. The labs you've done so far may have seemed repetitive, and it's because of this. So in those experiments you do, you usually have a table, right, where you have nothing but the data. Then you'll like write some sentences or a sentence or whatever, some words that basically just repeat what's in the table, which seems repetitive, and it is, but you're just putting it into words. And then you usually have a question where you take what you just wrote and use it to explain whether your hypothesis was correct or not. And that is what we're doing here. You've been trained to do this all semester. So this section is simply puts your data into words, right? So you're only going to say something like, all right, the pH started here, it ended here. This particular one increased or decreased the most. This one didn't change at all, right? Just the basics. And you only talk about the change in pH. You don't even talk about what it means. You kind of explained a little bit in the introduction what it should mean. And then later you're going to explain specifically what it means for your hypothesis. But yes, it's just putting into words what you have on the table. Make sense? All right, another common mistake. Again, this kind of falls under your lab report, should make sense to somebody who's not seen the experiment. When you're talking about your cuvettes, you want to write about them, about their condition, not their number. And that might not make sense, but in the lab manual, it'll say number your cuvette 1, 2, and 3, right? They all have numbers. But when you write about them, you want to say like the one that was on the ice had a starting point, started here, ended here. The one that was not on the ice started here, ended here. We don't care what cuvette number it is. Tell us what cuvette the condition was in. The one in the green light started here, ended here. The one in the white light started here, ended here. Does that make sense? Okay. Whew. Again, you're only putting into words what's in the table, and that is only a change in pH. Therefore, you should never mention photosynthesis or respiration. You're not explaining what the data means yet. You're just giving the data. Here's what the data is. And then later, you'll use the words photosynthesis and or respiration to explain what the data means. And when do you do that? You do that in the discussion. And again, this is very repetitive, but you've been trained to do this. And before I even read this, in short, what your discussion is you've done your results. Like I went from pH this to this. Now you're just explaining what those results mean in relation to your hypothesis. So here's my hypothesis. Here's the results. my hypothesis was supported or it wasn't, right? That's the big picture. Really quickly, let's run through this. Once again, you're going to need to restate the purpose of the experiment. So this is the questions this is the question we were asking in the experiment. This was my hypothesis. Yeah, you can read that example there if you want. But yeah, you're just going to restate the question. Here's the question. Here's my hypothesis. Again, you're going to restate it. But this time, instead of just restating your hypothesis, and again, instead of just looking at the data, you're going to breathe these things together. I hypothesized the red light would be the best light for photosynthesis. And as you can see from the results, the pH of the red light was the second highest. Therefore, my hypothesis was incorrect because it was not the best. Make sense? You're re-explaining, you're using your data that you talked about in the results section to explain if your hypothesis was correct or not. Another thing, and I know why this is to be a pet peeve of mine, but avoid the word proof. Right? You're not proving anything. This evidence of this data either supports your hypothesis or it doesn't. And it's not proving that you were right. And again, if this was a higher level class, we would talk more about statistics and I can explain why. But basically, that's it in a nutshell. We don't have enough data to say that it proved anything. Anyway, any questions so far? All right. And again, you've been trained to do this in your lab worksheets all semester. You will need to address the limitations of the experiment. Just like the first experiment we did, we talked about like, all right, technically we should all be barefooted. Technically we should have a better way of measuring the height. Technically we should make sure everybody's standing up the right way and shave their head, right? All these things, these flaws in the experiment, ways that it can be improved. You're going to need to write about that. As a matter of fact, it's a question in your lab worksheet, like it always is. And as always, this is a good hint to remind you guys, because I hate it when people, I always feel bad when people write, oh, there's nothing that could have been done. But if you haven't noticed, when we're doing experiments together, I'm talking about the ways we could help it, right? So when we're here together, pay attention to what I'm saying. Think, how can I make this better? What are some flaws in this experiment? All right. Any questions about that? All right. And then finally, for the discussion section, again, you've addressed your hypothesis, whether it was supported or not, using your data to explain it. And you talk about, all right, so here's the flaws, the flaws and limitations of my experiment. Then, as you would do in real science, say, how else can I test my hypothesis? So that's where you're going to write about you give a basic description of some other experiment you could do to test your hypothesis. And it should have nothing to do with algae beads. This is what we're doing for this experiment. You should think outside the box, think about plants, things like that. Yeah, that's it. Any questions about that? We're almost done. Figures and tables. You're already familiar with this as well. Figures and tables are what you're already doing in the lab or the lab worksheets. Figures, well, I should flop that. There's tables and figures. Figures are your graph. Table is your table. Your table, this is the easiest part, right? Just take the table that is in your lab worksheet and put it into your lab report. Of course, don't forget to label it, table 1, colon, and then describe the data in the table. And be careful with that. And I'm saying table 1 on purpose for this, because regardless of which experiment you did, for example, if you do the 5th experiment on the lab worksheet, that'll say table 5. But for you in your lab report, it is table 1, right? Because you only have that one table. Does that make sense? So don't let that confuse you. You'll lose a point for that. Include units. Of course, if you just copy and paste the table, then the units are already on there and you're good. And the only units we're dealing with are time and minutes. Minutes would be the unit, and pH. So pretty simple. Any questions about the table? Graph. Once again, you're going to make a graph for the lab worksheet. It's the same graph you can use for this lab report. The only difference is, for the lab worksheet, as always, if you want to, you can use this graph paper here and make a graph by hand. This is the one time, however, in this entire semester that I'm going to require you to make a graph on a computer. So even if you do the lab worksheet and you do a graph by hand, that's okay. But you're going to need to take that information and turn it into an actual graph. Does that make sense? Of course, I'm here to help you with that. Last week or 2 weeks ago, I forgot when, one of you asked a great question about like, hey, I know you said to use Excel or Google Docs, but can I use this other program? Yes, you can. It doesn't matter what program it is. as long as you do it on a computer. Again, you should label it. This is figure 1, regardless of which experiment you did. If this is your first graph, it is figure 1. As always, you're going to need to, after figure 1, you need to have a title that describes the data. And again, as always, your graph should include enough information that somebody who reads the graph without reading a lab report or the lab manual should understand what it means. That's your basic benchmarks for a graph. And again, when we do the experiments, just as usual, I'm going to draw you some hypothetical graphs of what it should look like. Anyway, any questions about this? All right, now this one's more of a formality. It's literature cited. For you guys, there doesn't have to be any literature cited, but you do need to still have a literature cited section that's labeled literature cited. And for you, if you only use the information in the lab manual or the lab worksheet, you just write that, right? Lab manual. But if you use your textbook, list the fact that you use your textbook. If you use some outside source, use it. And I'm not really concerned about the format because this is a 100 level class. Here's what I'm concerned about though. If you do decide to use outside sources, which is A-okay, actually encourage it. I want a working link to your source. So for example, if you're going to do an experiment about light intensity, and you want to look up more about like how photons work, and you do, and you include information about that in your lab report, and I read that, I'm like, what? What is that? Then I'll go to your literature cite section and we go, okay, well, they said according to this source, so I'll check that source. And as long as your source said it, you should be okay. So even if you write something that's incorrect, as long as you cited your source, I'll just say, hey, just so you know, that wasn't right, but you're not losing points because you did cite a source and your source did say that. Any questions about the literature side? Whew, we're done. 9:53. Oh man, I didn't do. It was such a rush. I didn't do attendance words. All right. So the last word for attendance will be example. Examples, last word for attendance. So if you get a screenshot, if you're watching the video, send the emails. That's it. And ask one question about the material covered in the lecture. I will see you all on Friday. Is that? I think so. If not, let me know.
Okay. Thank you.
Yeah. Thanks for that. I know that was an accident. I'm glad you did it because it's like a preview.
Rather be over prepared than under.
Yeah. Does anybody want to look at the stuff before we, if you want to? He's got it.
Algae beats.
Yeah, the algae beats, the materials.
Nobody cares.
What was the last attendance word?
What did I say? I just said it too.
Example.
Yeah. Yeah. Thank you.